We live in an hostile environment where we are surrounded by potentially dangerous bacteria, the viruses, and the parasite which may gain entry in our body, maybe in the air we breathe in or the water we drink. Yet, the body has an ability to resist these foreign substances that are potentially damaging to the body by the mechanism of immunity. So this immunity can be divided into two main types. One is an innate immunity or a natural immunity. It is present with us at all the times. Yet, whenever there is an entry of an organism, it becomes more efficacious. Then we have an acquired immunity, which is present only after an exposure to a pathogen. And the importance of this acquired immunity that it is highly specific to a particular agent that induces it. Although there is a considerable overlap between the innate immunity and the acquired immunity, we are organizing these into these many types just to understand them better. The innate immunity, as we said, is present from right the time of birth and works by providing various barriers against the infection. Whereas the acquired one is the immunity that is developed during the lifetime of an individual, and it is a specific type of immunity providing the resistance to many diseases that we encounter with. The innate immunity or a natural immunity is a non-specific system, although it is able to distinguish between a self and a non-self. That is possible because of the presence of MHC or the major histocompatibility complex, which labels the body cell as a friend, and hence they are tolerated by the immune system. It is not able to distinguish between the different type of agents or the pathogens, and therefore it is non-specific in nature, and it uses a pattern recognition receptor that recognizes different microbial structures and considers them to be a foe. So the non-specific defenses that are included in innate immunity, you can see it is the first line defense mechanism and the second line defense mechanism. In the first line defense mechanism, you can see the skin, mucous membrane, and the secretions. The second line being the phagocytic cells, antimicrobial proteins, inflammatory responses, and fever. And the third line of defense is formed by the adaptive or the acquired immunity. Now, a very sensible or the desirable way of protecting the body from the pathogen would be able to prevent them from entering the body by some barrier method. So, if we talk about these barrier methods, these barriers form the first line of defense. It may be the intact skin and the mucosa, which prevents the entry of the organism. And once the organism has entered and overcome these barriers, it is protected by the second line of defense, which are formed by antimicrobial protein, the phagocytes, and the other cells. Now, all the surfaces of the body which come in contact with the external environment are equipped either with a mechanical barrier or a chemical barrier to protect the penetration of the microorganism. The skin is one of the important mechanical barrier. It is essentially impermeable, that is protecting against any pathogen. Further, we have mucosa, which are present in a region which are not covered by the skin. For example, we have mucosa lining the mouth, then we have intestinal mucosa. These form the mechanical barriers. So what happens if there is a breach in the skin and the mucosa? Then what are the other mechanisms for the first line defense? Then we have the role of these surface secretions. The secretion which come from skin, it may be from the sebaceous gland, the sweat glands, the low pH which prevents the colonization of the microbe, or it may be the mucus, for example, in the respiratory tract, where the mucus is secreted by the mucosal membrane and helps to trap the microbes. Then we have certain secretions which are coming from our skin, for example, the sweat, the tear glands secrete the tears, and these help to wash out the toxic products and the microorganisms, and they also act to remove these bacteria or the pathogens by the fluid that is secreted by the skin. Now we have another important chemical barrier that is present in stomach that kills most of the pathogens And that is important because some of the pathogen enter our body through the gastrointestinal tract. Here in this slide, what do we see? We have different conditions in which there is a compromise of the first line of defense. The skin may be lost, maybe in the burns or the penetrating injuries, or there may be some damage to the mucosal lining as in respiratory tract infection. So whenever there is a compromise in the first line of defense, then what is the next step? The next step that protects our body from any infection is the second line of defense, and that is formed by phagocytes, the different type of antimicrobial proteins which are present. The inflammation, interferon, and even fever can act as a second line of defense. Now, a large number of pathogenic organisms and any inanimate particulate matters are removed from the body by the process of phagocytosis, which is nothing to eat up, and it is carried up by mainly the neutrophils or the macrophages. The neutrophils are the circulating phagocyte and most effective against pus-forming bacteria, while the macrophages are the tissue macrophages, and they are present in the different tissue components, and they wander through tissue spaces, the alveolar macrophages or the stellate macrophages which are present in the liver, or even microglia which is present in the brain. So whenever there is an entry of a pathogen, the phagocytes need to be mobilized. It involves different steps, for example leukocytosis, where a neutrophil enters the blood from the bone marrow, the margination, where it clings to the vessel wall, diaptesis, where it goes through the capillary, squeezes through it and enters the tissue space, and chemotaxis, where the neutrophil follows the chemical trail. And finally, the step of phagocytosis, where it consumes the pathogen and the cell debris, producing pus. The neutrophils are endowed with the capability to sense the infection, migrate to the site of infective organism, and then destroy the infectious agent. The inflammatory chemicals are released at the site of damaging tissue cells or the injury, or where the bacteria have entered the host defense mechanism. There are secretion of certain substances, maybe leukotrienes or complements, which mediate the first step, that is leukocytosis, which is nothing but the movement of neutrophil from the blood to the bone marrow. The next step is margination. When a neutrophil which is flowing inside a capillary approaches an area of inflammation. Here you can see in the figure, it becomes marginated, that is, it is attracted to the capillary endothelium and start rolling along its surface. So this is margination. Then is diaptesis, in which the neutrophil is trying to cross the capillary wall by insulating themselves between the endothelial cells. You can see here the neutrophil that is migrating to the tissue becomes elongated. It develops a broad head known as lamellopodium, and then a thin forked tail which is called pseudoflagellum, and it is trying to move into the tissue space. And these movements are guided by chemotaxins, that as neutrophils are attracted toward the site of infection by these chemicals which are released by these bacteria or damaging tissue, resulting in the process of phagocytosis. The phagocytosis steps first begins with the phagocyte which adheres to the pathogen or the debris. The green colored are the pathogens or the debris. Second, the phagocyte forms a pseudopod that eventually engulfs the particles, forming the phagosome. Now this phagosome, we have lysosomes which then fuse with the phagocytic vesicles to form phagolysosomes. The lysosomal enzyme digests the particles and leaving a residual body. And after that, the final step is exocytosis of the vesicles, removing all the indigestible and the residual material, and the pathogen has been rendered ineffective. The killing or the degranulation stage of a neutrophil is the oxygen dependent one. Here you can see where it is involving the oxygen for the generation of various substances or the oxygen reactive species. And secondly, we have an oxygen independent mechanism. Here you can see it is by the degranulations of the various substances which are present in the granules itself, for example lysosomes, which help to kill a pathogen. So the oxygen dependent bactericidal mechanism, as the name suggests, involves oxygen. Within seconds of stimulation of neutrophil, there is a sharp increase in the oxygen uptake, which is also known by the name of respiratory burst. So this increased oxygen uptake is utilized for the oxidation of glucose via hexose monophosphate pathway, and then resulting in the formation of reactive oxygen species. Here in this figure we see the formation of reactive oxygen species. So we can see the glucose which is taken up by for oxidation by hexose monophosphate shunt results in the production of NADPH and the reduced glutathione. NADPH reduces molecular oxygen to superoxide. You can see here the oxygen has been reduced to superoxide, and then this reduction of superoxide to hydrogen peroxide again occurs in neutrophil. It is catalyzed by superoxide dismutase, and then we have this reactive species which have been formed. However, the superoxide or the hydrogen peroxide does not have a significant bactericidal activity. So there is another myeloperoxide enzyme which help in the conversion of halides, that is chloride and bromide iodide, to form hypohalides, that is their acid form, which are the powerful microbiocidal agents. So you can see here. Next is an oxygen independent bactericidal mechanism where it works through lysosomal hydrolysis, defensins and the cationic proteins which are present in the granules of these neutrophils, and they are then released upon the entry of a microbe, and you can see the resulting mechanism has led to the death of these pathogens. So inflammation can be considered as a hallmark or a most important mechanism when we talk about the second line defense, and it is mediated by this phagocytic process. The cardinal signs of acute inflammation remains the redness, the generation of heat by the process of vasodilatation, the swelling in the region because of the alteration of the Starling forces, increased hydrostatic pressure, and then increased transudation of fluid into the tissue spaces, the pain which is associated along with it, and sometimes the inflammation may be so great that there may be loss or impairment of the there may be temporary impairment of the function. So when we talk about the second line of defensive mechanism, we also have these natural killer cells. They are large granular lymphocytes which are specially equipped to kill the virally infected cells and the tumor cells. So they are not phagocytic in nature, rather they attack the cell membrane and causes the cells to lyse. So some of the virally infected cells acquire a special glycoprotein on their surface, which then seem to announce to their neighbors that they are in trouble. The natural killer cells receive these signals and then they help to attach on the surface receptors and bring about the death of these cells before they could have multiplied. So, how are they protected? They have killer inhibitory receptors which interact with HLA class 1 molecules present on the normal cells, and once they interact and come in contact with the virally infected cell, the natural killer cells release the lethal substances which lead to the death of these infected cells, even killed before they are able to multiply. Then we have these interferons which are released by a virally infected cell. Infected lymphocyte released by maybe interferon alpha or beta or gamma. Although we have 14 different subtypes, but mainly it is by these. The interferons are released into the extracellular fluid, which then diffuse to form a protective ring of uninfected cells, thereby limiting the spread of infection. So you can see here, whenever a virus enters the cell, interferon genes are switched on, leading to the generation of interferon molecules. These interferon molecules then diffuse into the surrounding cell, and then they stimulate the turn on of a gene, that is antiviral proteins, by the surrounding cell. This antiviral protein blocks the viral reproduction. So it is basically forming a ring of cells which are protected by this virus, and it inhibits the protein synthesis by interfering with the process of translation and therefore promoting degranulation of the hair. So it is alerting your immune system so it can attack a virus or cancer cell. It helps your immune system to recognize various virus or cancer cell, stop them from growing and dividing, and keep the healthy cells ready for fighting the infection. So since virus depend upon the nucleic acid of the host for protein synthesis, interferons have a markedly inhibitory role on replication of the virus. It can be used to treat certain viral infections. It can be used in cancers, for example, hairy cell leukemias or lymphomas. And even it is being studied to see whether it can help in the treatment of COVID-19 infection by preventing the replication of this virus. We are familiar with the term of fever, in which there is abnormal body temperature, the systemic response to the invading organism which is not localized. So we have different steps involving this generation of fever. First, the leukocyte and the macrophages, which are exposed to the foreign substances, they secrete pyrogen. Next, the pyrogen act on the body thermostat that is present in hypothalamus, and it increases the body temperature. The shivering centers are activated, the heat loss centers are activated, as a result of which a person feels febrile and he has that shivering. So then, because of this, a negative feedback cycle will be initiated, and it will be responsible for restoration of normal body temperature. There will be heat loss, then again the blood will be carried to the hypothalamus, the thermostat, again resetting of the temperature, and this process of negative feedback loop will continue till the fever is decreased. So some of the beneficial role of fever are: first, it causes liver and spleen to sequester iron and zinc, which is needed by the microorganism. Then it increases the metabolic rate, therefore faster repair is there. It inhibits the pathogen growth because they may not grow in that high temperature. Then it increases the cellular metabolism, increases transferrin production, increase T cell production, and as I said, it enhances the repair process.